It's really it's a great pleasure to be here. I always love coming to India. My mother was born in India, and so there's a real family connection here. And she was actually born in Surat, which of course is now in Pakistan. And so I've always joked that I'm never going to tell the Indian immigration officials that my mother was born in Pakistan, but then to my horror, when I applied for the electronic visa, they asked mother's name, mother's place of birth. So I thought, oh my God, I'm never going to get in here. But anyway, there was no problem. So I came here without any glitches whatsoever, unlike Bill. So it's really a great honor to be here and talking to you. I'm not a theoretical physicist. I'm really an experimental biophysicist. But I'm one that really appreciates theory, and I've had the great fortune to work with some great theoretical physicists like Frank Eulicher, and actually currently with Yuhai Tu, whose name almost appeared on one of those slides there. So what I'm going to do today, I'm going to tell you what I regard as some of the what I think are the most important problems in biology, and mainly having to do with morphogenesis, the creation of biological structure, and then some of our work on that question. Okay, so I'm just hoping that the movie jinx is not going to affect me either, but let's try it. So what we're looking at here is it's a single cell alga actually, and these are cilia, and this alga, it needs to swim towards the light, and so it uses these microtubule based structures with motors all the way along the length in order to swim through the fluid. And I'm going to come back to this system towards the end of my talk. Okay. So let me start now with some of what I regard as important open questions about morphogenesis. That's the creation of structure in biology. And morphogenesis operates at many levels. So in development, how do organisms assemble? So how do cells form tissues? At the cellular level, how do cells get their shape? In other words, how do the molecules self-assemble into cells? And even at the protein level, how does protein structure, how is it encoded by DNA sequence? Now, these are forward problems. They're going from the microscopic to the macroscopic. But interestingly, the other big question, evolution, is it's a related but different question. That is, how does variation of genotype is selected by phenotype? And evolution is it's an inverse problem. So the macroscopic somehow selects the microscopics, and so this whole question of from morphogenesis going from the genome to the phenome, evolution is the process where it goes backwards in order to select for the right genome that will give you the phenome, which will circulate. It'll join the head to the tail and go around in a circle. Okay. I just want to give you a couple of examples of this beautiful morphogenesis in biology. This is the first 17 hours of development of a zebrafish, and what you can see is these cleavage divisions that are started from 2 cells here and going to roughly about 1 million cells. And what you can see over these 17 hours is the formation of the body axis with the head and the somites of the developing spinal cord. Incredible process. It's unbelievable to think how we're ever going to understand that. How do the cells know where and what type they should be in this organism? And another a set of questions, which for me is much more, it's more related to the work that I'm doing, is what determines cell shapes? I mean, here's a human sperm, here's a red blood cell, here's a keratocyte, a highly mobile cell. Here is a cell in our ear that senses sound and accelerations. Here's a section of a muscle fiber. Here is a mitotic spindle that is segregating the chromosomes shown here in black. the question here is how are the molecules organizing into these and building these structures which are thousands of times larger than molecular sizes. Okay, so here are what I regard as the key ideas if we're going to understand morphogenesis. We've got a bridge between length and timescales. Oligomerization is very important for polymerization, assemblies, liquid droplets, which are very important in the last few years. We've got, central to all of these ideas are self-organization and emergence, and that really is the key problem in biology, and we can say those words, but we really have no idea how to do it. Active matter, as we've just heard, is a key here, and that's because biological structure is active structures, and energy and material is constantly moving through biological structures. And finally, there's the mechanochemistry, and that is that you need to have some feedback between the shape or the morphology and the chemistry in order to be able to know when you've built something and how you can maintain it in that shape. And throughout all of this, the cells must be able to count, they must be able to measure length, and they must be able to tell time. So let me tell you the time scale, the length scales that we're interested in. We're going from the molecular scale, really at the protein level, down at the nanometer level, all the way through to protein assemblies, organelles, things like the mitotic spindle, and the single cells, and then into whole organisms, for example, such as this worm here. And the key organizational principle is this principle of active matter, and that's matter that is able to do work. It's able to extract energy out of a fuel, in this case ATP, and use it to drive some kind of directed motility. And this is the iconic Harvard video of a kinesin molecule pulling an organelle along a microtubule. And we'll come back and see some examples of this. Okay, and let me say that the energy currency here is ATP, and it's the hydrolysis of this of the gamma phosphate bond which is driving motility, and it's the hydrolysis of this bond, and each hydrolysis event gives about 25 kTs of energy. So it's depending on whether you think of the glass as being half full or half empty, it's a lot of energy. It's much larger than kT, but it's not that much larger than kT. Okay. So for the rest of the talk, I want to give you some of the examples of work that we've done that relates to morphogenesis. And I want to talk about Turing patterns. Of course, this is an old idea, but we have been able to make, I think, some new contribution to broaden this idea of the reaction-diffusion equation. I want to talk about mitotic spindle length control, so how cells use an active ruler to know how large cellular structures are. I want to tell about how the cilia beach or the flagella bead is generated, what I showed you in the first movie. And then finally, I talk about the branching morphogenesis of neurons, which is another microtubule-based structure process. So all of these are microtubule-based processes and driven by motors, which are my favorite molecules. Okay. So how do you make patterns in biology? So the classic work of Turing, you can't overemphasize how important this is in biology. And Turing's morphogens, he really coined a phrase in the 1950s, and it really dominates the thinking in developmental biology now, the concept of the morphogen. And it is amazing that it wasn't until around about 1980, about 30 years later, the first morphogen was discovered. Now, Turing was thinking about how the zebra got its stripes. And his proposal was that there were two flavors of morphogen, an activator and an inhibitor. And the idea was that the activator diffused slowly, and so it acts locally. And whereas the inhibitor diffuses quickly, and so it acts long range. And with these, just these simple two states, he was able to show that you can get instabilities that give rise to patterns and all kinds of dynamic phenomena. And this was formalized by Gira and Meinhardt. And I'm not going to go through these equations, but to show that you can get spatial patterns out of these very simple equations. Now, Meinhardt was interested in trying to understand the development of the fly, and it was in the fly that Janni Nussler and Volhard discovered the first morphogen. And so Meinhardt was interested in trying to understand how the stripes came about in the fly. But I'd say that whether this is probably not a reaction-diffusion equation, because the diffusion is too slow to be able to generate a pattern which is as large as this, on the order of a millimeter in length. But rather, it's been incredibly productive for looking at patterns in, for example, bacteria on a much smaller timescale. This is the MinD protein oscillating in a bacterium, and where the concentration is the largest is where will determine where the cell division takes place. And remarkably, it's possible to purify just two proteins out of these bacteria, put them back together under the microscope, and in the presence of ATP, you get these beautiful chiral waves. And so these sorts of Turing-type patterns, I mean, they really do exist in biological systems, and very, very important for example, bacterial division. Okay, so the idea with these MinD proteins is that they measure the length of the bacterium. So the bacterium grows, and it grows to a certain length, and at that length, it's a length at which the MinD oscillator will tell it to divide. And so what specifies that length? And for a reaction-diffusion system, if you have a diffusion coefficient and some kind of reaction that degrades the molecule, so if there's a source here and it can diffuse away from the source and get degraded, it defines a length scale given by this formula here. Now, it turns out that this is rather limited, but if we're allowed to have active materials, then we have many other different possibilities for length measurement. So, for example, if we have advection in diffusion, where there's directed motion in this way, but there's diffusion in that way, there will be a length given by this formula here. And likewise, if there's advection moving in this way, followed by, from a source here, and degradation, then there will be a gradient given with a length determined by that. So what you see is that by having motors that can give rise to directed motion, you can get different length scales in systems. Now, the reaction diffusion is what sets the length scale in Turing patterns, but in other systems, for example, in those hair cells, in the cells in the ear, the stereocilia, the length is determined by motors that most likely use an advection diffusion mechanism to set their length. And in cilia of the type that I showed you with chlamydomonas, they likely use an advection reaction type mechanism for setting their length, although these mechanisms still have to be worked out in detail. Now, what I want to do now is tell you about that we discovered a new and very surprising mechanism for specifying length. So let me tell you about that now. So kinesin is a motor, and I showed you that picture of kinesin walking along a microtubule. But it turns out that there's a big family of kinesins, and these kinesins do all sorts of things. Many of them are motors, but others do other things. And this particular kinesin, called kinesin 8, it is a motor, moves along microtubules, but it also depolymerizes microtubules. And that, when you have a motor that depolymerizes, you get very interesting properties. Now, the cell biologists had shown that this kinesin 8, it regulates the length of the mitotic spindle, and the standard assay for that is that here, for example, is a cell in the fruit fly Drosophila, and it has a mitotic spindle of a certain size. Sorry about that, that's about 10 microns. And if you knock out or knock down this kinesin 8, then the mitotic spindle gets larger. And if you overexpress the kinesin 8, the mitotic spindle gets shorter. And this is seen in Drosophila cells, but it's also true in yeast cells and human cells. A very universal finding. So the question is, how does kinesin 8 regulate the length of the spindle? So to answer that question, we set up this, what we call a depolymerization assay. It's a microscope assay where we bind a microtubule to a glass surface. We illuminate with a laser so that we can use total internal reflection fluorescence microscopy. And then we add our motor of interest. In this case, it would be the kinesin 8. And when we add kinesin 8 in the presence of ATP, what we see is the following thing. So here are some microtubules. They're bound to a microscope slide. Then we add the motor, and what we see is the following. Boom. Microtubules get shorter. So this motor is depolymerizing the microtubules. So it's actually eating up the track that it's walking on. So, and we call that a depolymerase. Now, if we plot the lengths of the microtubules, what you see is that the microtubules shorten, but what you see is that the longer the microtubules shorten faster than the short microtubules. And I just plotted here is an example where the depolymerization rate is plotted against the initial length of the microtubule. And what you see is that the depolymerization rate increases with the length of the microtubule. So it's a length-dependent depolymerase. Now, that begs the question, how does a little motor that's only about 10 nanometers in length know to depolymerize a long microtubule that's 1,000 times longer than its length? How does it know to depolymerize this microtubule faster than this microtubule here? So in order to answer that question, we turn to our single molecule techniques. And what we're able to do is, if we now fluorescently label the motor green and the microtubule red, what we found is that the motors are what we call highly processive. So what that means is that the motors, they diffuse down from solution and they bind to the microtubule, and then they move all the way along the length of the microtubule. And what you're seeing here is these motors appearing, and then they all move to the end of the microtubule. And then what we showed was that when they get to the end of the microtubule, they remove just a single tubulin dimer, the subunit of the microtubule, from the end of the microtubule. So what happens then is what we call the antenna model. So motors bind randomly along the length. They move to one end, the so-called plus end of the microtubule. Microtubule is the polar structure. And then they just remove a single tubulin dimer from the end. And this combination of a processive motor and a low stoichiometry depolymerase gives you this length-dependent depolymerization, because the longer the microtubule, the more motors land on it, the more that get to the end, the more tubulin dimers get taken off the end of the microtubule. And so this explains the length dependence. So our idea then is that we've got this negative feedback which sets the length of the microtubule. So if the shrinkage rate increases with length, the growth rate is constant, then at where these curves intersect is a stable point. And if the microtubules grow longer, then the shrinkage exceeds the growth, and if they're shorter, then the growth exceeds the shrinkage. So it's a stable system. And so this is our idea then for how this motor can play this role as regulating the length of the mitotic spindle. And we think that this is a general mechanism that can explain the length of the mitotic spindle, but also the length of the hair cells, and also the length of cilia as well. And so so I was just going to say that the idea is that these motors can perform these mechanical computations in order to calculate the sizes of things. Okay. Now, it's very interesting to think about the energetics of this mechanism. Now, what's really happening here is that in order for this little motor to measure the length of something big, what the motor's really doing is it's pacing out the length of the microtubule, and it's using the energy from ATP hydrolysis to act as a ruler for the length of the microtubule. But the thing is that every step that it takes costs a molecule of ATP. So that means that because each step costs a molecule of ATP, there are literally thousands of ATP molecules consumed for every tubulin diamond that's taken off the end of the microtubule. And when you consider that you need a large number of motors in order to precisely regulate the length, this is, turns out to be a very expensive way of measuring something. And it really begs the question, and it's a question that we've been fascinated in not just this system but in other systems as well, is just the general question, how much do cells pay for information? Because we know at one level, for example, in the synthesis of ATP, cells are incredibly energetically efficient. But when it comes to signaling and getting information about itself, they're just reckless in the amount of money that they spend, and we have no idea about what really the worth of information is in biological systems. Okay. I want to now go back to cilia and our trying to understand this beautiful bending pattern of cilia. And what we found is, and what we believe is that there's the cells can sense this curvature of the cilia, and that feeds back onto motor activity to regulate this beautiful beating pattern. Now, I should say that this is work that is a long-term collaboration with Frank Ulish, a succession of students, and so I want to say that what I'm going to tell you about now is work from a theoretical student of Frank's, Pablo, and an experimentalist, FICO, in my group. So, cilia are found, for example, in these protozoa, but they also drive sperm motility, and they're also very important for moving fluid within the body, in the brain, in the lungs, and they're also very important in development. Now, the cilium, these are some pictures from Leeuwenhoek. It was what he regarded as his greatest discovery. And if you now, many years later, 300 years later, look through at a section, you'll see that they're made up of microtubules, so-called doublet microtubules, and then in between the microtubules are these dynein motor proteins, and these are located all the way along the length of the cilium, and these drive sliding of the microtubules, and then that sliding gets, due to constraints of sliding, gets converted into to bending. So the question is, this is a human sperm cell. Question is, how are these dynein motors coordinated? So in order to answer that question, the first thing we need is some model and some ideas about how it might work. And of course, this is something that Frank has worked on for a long time. And basically what they did back here, they formulated a 2D model of the cilium. And I don't want to go through it in detail how the projection is worked, but basically you take the three-dimensional section and you project it down into just the two filaments, and you take motors which on opposite sides of the cilium, one set of motors bends it in one direction, the other set in the other direction, and you collapse that down to motors with opposite orientation between these two filaments. So these are the features of the model: inextensible filaments, oppositely oriented motors. There's a basal region which restricts sliding, but the tip is free, and there's a constraint that the two filaments have a constant spacing. So that's the mechanical model, and you can write down an equation of motion, and this was done by Ken Muchin in 1958, in which he balanced the sliding forces against the viscous forces and the bending forces, and these are the various parameters, but basically F is a force and psi is the bending angle. The A is the diameter, this is the drag coefficient, and that's the bending stiffness of the structure. Now, model like this, problem with the model like this is that we have to know what the force is that's driving it, and we don't know what the force is. And the idea for developing a theory of motion of this thing is to propose that the force depends on the angle or the sliding or whatever, and then if we can have a force which depends on angle, then we've got an equation and we can solve that equation. And I'm just going to give you the curvature feedback model because this is the one that works. Other models don't work, but basically the idea is that the dyneins between the filaments, when the curvature has one sign, they will detach from the filament. And so what we can do is we can write the force depends on the instantaneous curvature, so that's the derivative of the tangent angle, and also to the rate of change of curvature. So we have a system where the force can depend in phase and out of phase with the curvature. If we now substitute this force equation back into the last equation, then we get this fourth order complex differential equation, which we can then solve with the appropriate boundary conditions. And then the solutions are then predictions that can be compared to measurements. And we just have really a single control parameter, which is this beta, which corresponds to the real and imaginary components of the curvature sensitivity components. We can compare this with the shapes. What we can do is we can knock the cilia off the cells, and then we can reconstitute the motion. We can measure the motion of individual cilia, and here's an example of that, and we can track the shape very precisely. And this is what we're doing here. The red is the base, the black is the midpoint, and the green is the tip. And so you can see you can get these beautiful patterns. And what I like about this slide is that when I was a kid, my favorite toy was a spirograph. And this just gives us gazillions of spirographs. Very nice system. We've got it all in the computer. We can put it all back together again. And the bottom line is that this curvature feedback can give us predicted shapes which match very well with the experimentally measured shapes. And we can do this in wild-type cells. We can also do this in, for example, mutant cells that have got symmetric beats, and we can change a lot of other parameters as well. So this is really a very well tested theory, and this curvature feedback works very well. Okay, so in the last minute, I'll want to tell you about our recent, and we've only got an archive paper on this, on branching networks of neurons. And this is interesting for us as well because the dendritic tree of a neuron is composed of microtubules as the structural elements with motors that are carrying materials in and out of the processes. Now, we're using these mechanoreceptors that tile the surface of a fly as a model system. They're highly branched cells. They've got over 1,000 branches. And it's a a similar but related problem to, but different to other branching problems in biology, for example, in the lungs and in the vascular system. But we're talking about branching in single cells, whereas this is branching in tissues. The fundamental question is, how are branches formed? And how do you get this structure which forms over about 4 days? And what are the molecular and cellular rules? And so, this is early, it's early stages yet, but the first thing is you've got to look and you've got to observe what goes on. And the great thing about this fly system is we can look right into the fly and look at the branching mechanism as it actually takes place in real time. And this is in the embryo. Here are 2 of those cells that are at a small stage. And what you see, it's absolutely unbelievable. This is a very, very highly dynamic process. And what you see is that these branches, they're growing out, they're shrinking, they're growing back out again. It's an unbelievably dynamic process. And this just gives you some examples of the sorts of things we see. We see tip growth, branching. We see tip retraction, and we can actually have retraction through several levels of branches. And there's also this self-avoidance phenomenon where a branch will grow out, it'll hit another branch, and then it'll retract again. And so what we're trying to do is try to figure out what the molecules are doing there, and also how these kinds of local rules can give rise to a global branching pattern. And this is an example of some of the sorts of models that we are generating, computational models, is by a graduate student, Olivier. Okay. So let me just come back and just summarize now. I mean, what do I see as the big challenges? The challenges are, what are the molecular mechanisms by which cells measure length? They can sense their shape, they can count, they can tell time. These are all emergent properties. How does that work? And how much does it cost to create these self-organized systems? And then finally, what is the role of evolution in generating morphology? And to say, the interesting thing about evolution, that it acts at the phenotype, which is the emergent property, but yet the variation acts at the molecular level. So it seems to go, it goes backwards. And that's all I've got to say. Thank you very much. And you can visit the lab on Facebook. So thank you very much. Give him 2. There you go.
No, so you were talking about this question of how much does information cost. So people often tell you that, look, ATP is very abundant in the cell, so this isn't an issue. People informally say something like this. And how is that right? Is that not to be taken seriously?
That can't be right, and I'll tell you why it can't be right, and that's the following. If you look at the synthesis of ATP through oxidative metabolism, the efficiency of that process is 90+%. That means that we can harness roughly about 90% of the energy from glucose and put it into ATP. Likewise, for example, ion pumps that pump sodium and potassium across membranes, they essentially work at thermodynamic equilibrium with the ATP potential, meaning that they can pump ions with about 90+% efficiency. So muscle is about 50% efficient. So it's clear that energy is very, very important for the organism. But when it comes to signaling and controlling what happens in the cell, the amount of energy that's spent it's truly staggering, and we really have no idea about how to even set the order of magnitude of that energy. Not even, is it 10, 100,000, whatever? How much, what's the market rate? I mean, what is the economics of that? And I think we just don't know. And that, in my view, is really one of the the fundamental questions, and it's expensive. They spend a lot. It's nothing like the Landau limit. You know, there's none of this KT per bit of information. It's way, way higher than that.
Kind of halfway through your talk, or maybe in the first half, you showed how, with very simple ingredients, you can get characteristic length scales.
Mm-hmm.
And one of the cases you talked about was advective diffusive system.
Yes.
So the velocity and the diffusion constant give you a length.
Yep.
But I think about it, it seems to me that the advective diffusive system, it'll have a length scale, no doubt, but it'll give just an exponential signal with that length scale. I don't see where you get the oscillating pattern out of that.
Yes, this is with Turing patterns. That's the question with... No, I mean, with the Turing patterns, they are, you know, reaction and diffusion, and basically what happens is that the inhibitor diffuses away so that you can get that, you know, so if you look at one cell, if there was no diffusion of the inhibitor, then the inhibitor would build up and inhibit the process, but it can diffuse away, and so you can get sort of autocatalysis there. And but then what happens is autocatalysis. But the inhibitor eventually inhibits the other cells, and so they now no longer they will no longer inhibit that, and you can get a pattern in that way. With advection, then it's a good point. You can use advection to have enhanced diffusion, for example.
But it doesn't make an oscillating pattern. Is that the point?
You know, it depends on, you know, like for example, in the cilium, we've got a driven system with a directed motor giving rise to a pattern. It's a temporary, you know, it's the wavelength of the beat of the cilium. So, you know, I think in each case you can do it. You know, you can solve. You know, you can find a solution to that.